Okay, cool. AQA A-level biology topic 3 completely patterned. Yeah, I'm gonna go through all of exchange of substances. Yeah. Alright, let's start. Okay, so let me explain exchange quickly. Yeah. So single-celled organisms have a large surface area compared to their volume. Yeah. That's obviously due to their small size because they're small. They got a small volume. Calm. A large surface area allows for efficient gas exchange. Yeah. The reason for that is obviously because if you got more area, you got more room to interact with the gases. You know what I'm saying? Okay, cool. As organisms get larger and larger, yeah, they become multicellular and their surface area to volume ratio decreases, yeah. The reason it's decreasing is because the volume is increasing. So because the volume is getting larger and larger, the ratio is gonna decrease, you get it? Okay, cool. Larger organisms, yeah, face challenges in efficient exchange due to the decrease in relative surface area. To overcome this, multicellular organisms develop specialized exchange surfaces such as lungs, gills, and intestinal walls, yeah. So features of an efficient gas exchange system. So these three things have to be there for an efficient gas exchange system. Yeah. First thing is large surface area. So surface area can be increased, yeah, using root hair or folded membranes, yeah, and that's going to provide a larger surface area, which means more efficient exchange system, yeah. Another thing is thin exchange surface. So obviously you need to provide a short diffusion pathway. In order to do that, the wall's got to be thin, and if it was thick, yeah, then it's not going to move through that. You get me? So it has to be thin so that it can move through it fast. And it also got to have a good blood supply, yeah, so good ventilation, yeah, so that's gonna allow room for the blood to come through quickly, get ventilated, all of that. You get me? Cool. I'm gonna explain why increasing the size is gonna decrease the surface area to volume ratio. Yeah, the reason for that is because if you start bare small like this, yeah, you obviously see that we've got one on each side, 1 + 1 + 1 + 1, whatever is gonna equal 6 as the surface area. So we've got quite a large surface area there. Safe. But the volume of which this surface area is covering, yeah, is a very small volume because just 1 × 1 × 1, yeah. So therefore we got 6 surface area, yeah, for one volume. So it's 6 to 1 ratio. So it's a very good high surface area to volume ratio. So therefore the fusion is going to be efficient through that. But if you increase the size, yeah, surface area increases to 24, yeah, that's calm. But volume increases to 8. So the surface area to volume ratio between them is only 3 to 1. So therefore the ratio decreased here. The gap between them two numbers decreased, you get it? We come down here, yeah, we increase the size again, 54:27, yeah, so that's a 2 to 1 ratio now, so it's quite shit. So you see the situation, as you increase the size of the organism, yeah, the surface area to volume ratio is going to decrease here, which means that it's going to be less efficient, isn't it? Okay, cool. That's why you need specialized exchange systems, yeah. So if we look here, yeah, we got fish here. Fish have a small surface area to volume ratio, yeah, obviously because they're quite big, yeah. And they also have an impermeable skin for protection, yeah, but that also means that it can't gas can't come through that skin. You get me? Okay, cool. They require a specialized gas exchange surface, therefore, yeah, which are the gills. The gills are obviously in pairs of four, yeah, are 4 pairs, yeah, of gills, which are supported by an arch. The arch is just like the bone behind the gill, yeah. Okay, cool. On these arches, we've got gill filaments, yeah. So this is the actual gills themselves, yeah, which have little lamellae, yeah. Lamellae participate in gas exchange. Blood and water flow across the lamellae in a counter-current direction. I'm gonna talk about that in a minute. And they maintain a steep diffusion gradient. This ensures maximum oxygen diffusion in deoxygenated blood, yeah. You gotta say about when you talk about maintaining a steep diffusion gradient, you gotta say maintains it across the whole length of the lamellae, yeah, or across the whole length of the gill filament, yeah. Okay, cool. Gill filaments are held apart by water flow, causing them to stick together out of water. So that's why fish can't breathe outside of water, is because these gill filaments need to be held apart by that water flow. So it has to have that water flow consistently. You get me? Okay, cool. Fish cannot survive for long periods of water due to this dependence on water flow for gas exchange. Nice. So the reason they need water is it's because water flows through the mouth like that, opens the gills, yeah, and when it opens the gills, it moves through here. And these are the gill arches I was talking about, the bone, and it has these gill filaments on them, yeah, the red things, yeah. And these gill filaments have got lamellae coming the other way like that. And these lamellae, they look like this. They got deoxygenated blood, that blue thing, moving across the left like that to the oxygenated blood. It becomes oxygenated, and the water is the blue thing, the blue arrow that moves that way. So as you see, they're moving in opposite directions. The blood's moving where my mouse is now, and the water's moving where my mouse is now. So therefore, this shows you as well. There's always more oxygen inside the water compared to the blood, only slightly more. So there's always that diffusion gradient that's maintained here across the whole lamellae and therefore across the whole gill filament. Yeah. Alright, calm. So this is what I'm talking about, the co-current flow. If it was co-current, so if water and blood was moving the same way, let's say that the blue thing is the blood here and the red thing is the water, Yeah, we're gonna start 100% oxygen in the water. It's gonna move to the blood. Yeah, so therefore the blood's gonna increase in oxygen until 50%, and the water's gonna decrease in oxygen until 50%, and then it's gonna reach a standstill. It's gonna reach a plateau. You get me? They're gonna balance each other out, so it's gonna be 50-50 now. So therefore, it's the diffusion gradient's not been maintained. Yeah, that's why it's got to be counter current flow, where the water's moving like that and the blood's moving like that. Therefore, the water's getting deoxygenated slowly and the water's getting oxygenated slowly, but there's always more oxygen in the water, so therefore it's gonna be, you get me? Let's say down here is 10%, that's gonna be like 15%, so therefore it's gonna go down. Here's gonna be 20%, that's gonna be 25, gonna go down. You get it? Cool. Ventilation, yeah. So ventilation is the actual way that the water moves through the fish. So ventilation is necessary for the fish to maintain a continuous one-way flow of water, yeah. So it starts with the fish opening its mouth, which obviously allows water to flow into my man. Yeah, then the floor of the buccal cavity lowers. Yeah, so the buccal cavity is like the floor of my man's like mouth. Yeah, and that's gonna move down. It's gonna open wide. Yeah, like you're at a dentist, and it's gonna increase the volume and draw in more water. You see what I'm saying? Once the mouth is closed, yeah, the buccal cavity floor raises. Yeah, creating pressure. So once the mouth gets closed, yeah, it creates some pressure here, which obviously moves the water through the gills. The pressure differences force water over the gill filaments, yeah, which obviously is what we need. And then you got the upper column here. The upper column here acts as a valve and pump here. So it acts as a pump that pumps the water through in and out of the gills, yeah. You get what I'm saying? That's the upper column them there, yeah. The little gill cover or whatever, yeah. Okay, cool. Insects here. So obviously upper column is like a bit of an extra thing, but you can use it in the, you get me, you can use it in the finale, yeah. In the thing in the essay, yeah. Alright, cool. Insects lack transport system, yeah. So obviously they don't have a transport system. So oxygen has to be directly delivered through the respiring tissue. So the air has to come here through the spiracle, yeah, through a little hole inside the wall of the inside the body of the insect, yeah. It's got to come through the trachea, a little windpipe, yeah, go through the tracheoles and get delivered directly to the cells. So this is accomplished through spiracles, yeah, small openings connected to tubes called trachea or tracheoles. Alright, safe. These tubes extend throughout the insect's body, yeah, supplying it with necessary gases, yeah? Cool. Gases move in and out through diffusion, mass transport caused by muscle contractions and changes in tracheal volume, yeah? So what the insect can do is it can actually like scrunch up here and it can actually like move its body in a certain kind of way, yeah? To basically force air into itself here. So like it can move around its like stomach here to help air get forced in through the spiracles here. Obviously, the other way that it's done is obviously just through diffusion. The air is gonna diffuse through that spiracle. It's gonna go inside directly to the cell. You get me? Alright, plants. Plants have evolved various adaptations in their leaves to facilitate efficient gas exchange. Yeah, one thing is a stomata. Yeah, so stomata are small. They're like the spiracles. Yeah, they're small pores on leaves. Yeah, which enable gas to enter and exit. The abundance of stomata ensures that no cell is far from one stomata. So every cell in the plant is near stomata. You get me, and that reduces the diffusion pathway. Yeah, so therefore I've got bare cells here. I've got stomata there. So therefore it doesn't have to travel that far to get there. Additionally, leaves contain a lot of air spaces here that allow gases to circulate and reach photosynthesize and mesophyll cells easily. Yeah. Okay, cool. Humans, yeah. So humans have lungs, yeah, which is located in the chest, and they're paired, they're lobed, and they have a large surface area, yeah. So the rib cage surrounds and protects the lungs, yeah, of course. There's also a lubricating substance that prevents friction between the rib cage and the lungs during inflation and deflation. Obviously, when you breathe in, your lungs are going to get inflated, so that's going to cause a bit of friction, yeah? So you have to have lubrication there, you get me? Okay, cool. External and internal intercostal muscles, yeah, located between the ribs, contract to raise and lower the rib cage, yeah, facilitating breathing, of course, yeah. Calm. Because obviously you can't just breathe and the rib cage not move, yeah. It has to slightly raise and lower. That's why you need the external and internal intercostal muscles, yeah. The diaphragm structure separating the lungs from the abdominal area plays a crucial role in breathing. You'll see it in a minute. Okay, cool. So let's go through respiration, yeah. So respiration is how you breathe air into the lungs, yeah. So air enters the respiratory system through the nose. So it enters through the nose, yeah, unless you breathe with your mouth, yeah, and it comes down into the trachea. Then after it moves through the trachea, it's gonna move into the bronchi. Then after it moves into the bronchi, which is these little things that part away from each other, it's gonna leave into the bronchioles, which is inside the lung now, yeah. And then it's gonna go to the alveoli. So what you're doing here is just locate, because you got a low surface area to volume ratio, yeah. You got a lot of volume, yeah. All it's gonna do is trying to localize that air into like a small area that has a small volume. Imagine how small of a volume the alveoli has here, and imagine the surface area of the alveoli. Yeah, so that's what it's trying to do. So these airways are kept open by rings of cartilage, of course. Yeah. Okay, cool. Bronchiole is narrower than bronchi. Yeah, they lack cartilage and are primarily made of smooth muscle and elastic fibers. You need to know about that. Bronchioles lead to clusters of alveoli, which are small sacs filled with air where gaseous exchange occurs. Yeah, so of course in these alveoli, yeah, you got the capillaries that are wrapped around it. Yeah, and these capillaries obviously have the blood, so the air is gonna come through the alveoli and just diffuse into the capillaries. You get it? So during breathing, yeah, the lungs inflate and deflate within the chest cavity. External and internal intercostal muscles, along with the diaphragm, aid in raising and lowering the rib cage. I'm going to go into more detail on that in a minute. You don't need to write this down. Facilitating air movement in and out of the lungs, yeah. Gaseous exchange transpires across the walls of alveoli, ensuring the exchange of oxygen and carbon dioxide between the lungs and the bloodstream, yeah. Alveoli adaptation. So as you see, the deoxygenated blood comes from the heart like that. These the alveoli, yeah, are filled with air, so of course it's going to move across. It's very thin, so it's got a short diffusion pathway. It oxygenates the blood and the blood returns to the heart, yeah. So alveoli are extremely thin, approximately 1 cell thick, and are surrounded by capillaries, yeah, minimizing the diffusion pathway for gases, yeah. The continuous blood supply by capillaries ensures a constant steep concentration gradient, optimizing gas exchange efficiency. Calm. And the abundance of alveoli provides a vast surface, minimizing the area available for maximizing the area available for gas exchange. Yeah. Alright, cool. So these are adaptations of alveoli. Yeah. Structures. Yeah. So cartilage. So you need to know certain structures within the thing. Yeah, and how they play a role. Yeah. So cartilage. Yeah. So it's involved in supporting the trachea and bronchi, not the bronchioles. They don't have cartilage. Yeah. And plays an important role in preventing the lungs from collapsing in the event of a pressure stop during drop during exhalation. Yeah. So if it wasn't for that, then during exhalation your lungs would collapse. Yeah. Ciliated epithelium, yeah, epithelium, yeah, present in bronchi, bronchioles, and trachea, and involved in moving mucus along to prevent lung infection, yeah. So of course the mucus is gonna move along and trap bacteria, and that's what helps your lung from not getting infected, yeah. And obviously that's why sometimes you have to clear your throat because that's the mucus that's coming through. Goblet cells, yeah, these are the things that actually make these mucus, yeah. So the cells present in the trachea, bronchi, and bronchioles involved in mucus secretion to trap bacteria and dust to reduce the risk of infection, yeah, with the help of lysozymes. Obviously, you know lysozymes from topic 2, yeah, and you know what they do. Alright, cool. Smooth muscle, yeah. The ability to contract enables them to play a role in constricting the airway, thus controlling its diameter as a result, and thus controlling the flow of air into and from the alveoli. So these smooth muscles, they just contract in certain ways to move the air directly to the alveoli. Yeah, you get it. Elastic fibers, yeah, stretch when we exhale and recoil when we inhale, thus controlling the flow of air. So they're the same as smooth muscle basically, but they recoil and shit. Okay, cool. Inspiration, yeah. During inspiration, the external intercostal muscles contract. So for inspiration and expiration, you need to know which muscles do what, yeah. So inspiration, yeah, external contract, internal relax, diaphragm contract. Always the external and the diaphragm does the same shit, yeah. So if external's contracting, yeah, then the diaphragm's contracting as well. Remember, the diaphragms are main state is when it's up. So contracting means it moves down, yeah. So what happens is the external muscles contract, whereas the internal relax, yeah, and as a result, it causes the ribs to raise up. The diaphragm then contracts and flattens, yeah, and this causes air to obviously move in and down, and the volume inside the thorax, yeah, which is this whole thing here, the thoracic cavity, yeah, increases, and the difference between the pressure inside the lungs and atmospheric pressure, so the pressure outside, creates a gradient, which obviously causes more air to be forced into the lungs, yeah. So you need to memorize that whole thing there, yeah. And for expiration, it's the opposite way. So in expiration, the internal intercostal muscles contract. So before it was the external that was contracting, now it's the internal that's contracting, yeah. And then the external is gonna relax. So external muscles relax and therefore lowering the rib cage. I told you the diaphragm does the same shit as the external, yeah. So the diaphragm also relaxes and hence it raises upwards, yeah, because that's its base state is to relax upwards, yeah. And that therefore obviously pushes air out of the body, yeah. This action in combination to decrease the volume inside the thorax, therefore increasing the pressure, forcing the air out of the lungs, yeah. Okay, cool. Spirometer, yeah. Spirometer is a medical device used to measure lung function by assessing the volume of air a person inhales and exhales, yeah. It consists of a chamber or a bell that is connected to a tube. So just got a little situation like that. You clip the nose so that air can only move through one source, and yeah, you just exhales into it. When a person breathes into the tube, yeah, the changes in air volume within the chamber are recorded. This information helps evaluate various indications of lung function, yeah, so they're trying to see if your lungs are healthy or not, and it will see, you know, your lung capacity, your volume, and your flow rates. It's used to obviously also diagnose asthma, yeah, so that's the asthma situation here with the canister and all that. Calm. Key terms, yeah. Vital. So you need to know a couple key terms here that are important in the situation. They might ask you to define these, that sort of thing. Yeah. So we've got vital capacity. Yeah. The vital capacity is the maximum volume of air that can be inhaled or exhaled in a single breath. Yeah. So if you go like ha, yeah, that's the vital capacity there. You see that? Okay, cool. Varies depending on obviously gender, age, size, as well as height. Yeah. Tidal volume, yeah, the volume of air we breathe in and out at each breath at rest, yeah. So the tidal volume is just like how the volume of what you normally breathe in and out, just regularly, yeah. And then the breathing rate is the number of breaths per minute and can be calculated from the spirometer trace by counting the number of peaks or troughs, yeah. Troughs are just peaks, but at the bottom of the wave, yeah. Alright, safe. The volume of air which is always present in the lungs is known as the residual volume. So sometimes there's always going to be a little bit of volume of air that's still just left over. Yeah, that's the residual one. You see what I'm saying? And during exercise you can breathe in more air than usual, yeah, by using the inspiratory reserve volume. So this is extra air that you can inhale after a normal breath. So during exercise you can use that reserve volume to inhale a little bit more breath instead, yeah. And same way you can also breathe out more air by using the expiratory reserve volume, yeah. Okay, cool. Digestion, fam. Digestion. So you got a couple situations with digestion. Yeah, you got carbohydrates, which are digested by many different enzymes. First one you need to know about is amylases in the mouth. I'm using the plural term here because there's different types of amylases. Yeah, and they're in the mouth and they digest larger polymers. Yeah, so you eat the bread, it's got the polymer, polysaccharide. It's going to be digested by amylase straight away. That's why it gets sweet. Yeah, maltases in the ileum break down monosaccharides. And sucrases and lactases break down the disaccharides, sucrose and lactose, respectively. Yeah. Okay, cool. Let's talk about lipid digestion. I'm gonna talk about lipid digestion very briefly here, and we're gonna move on to it in more detail in a minute. Yeah. So lipids are digested by lipases. Yeah, which obviously hydrolyze the ester bond between the monoglycerides and fatty acids. Yeah. If you know about topic one, you know about the ester bonds that's formed between monoglycerides and fatty acids. Yeah. And obviously, what lipases do is they come and they hydrolyze that, so they break that bond. Yeah. And before being broken down in the ileum, lipids are emulsified into micelles by bile salts released in the liver. So micelles are like little like bubbles of fat basically, and that's called emulsification. And this emulsification just increases the surface area and speeds up the chemical reaction. So you have a piece of fat like that, it will come to the bile salt because fats are kind of hydrophobic in a way. They're lipid soluble. Obviously, they're lipids themselves, yeah. Then they're not gonna mix with water. So the bile salt has a hydrophobic side and hydrophilic side, yeah. So the hydrophobic side binds to the fat like that, and it wraps around the fat, which increases the surface area because it's gonna break that down, and now you got even more surface area on both of them. You get it? And that's called emulsification. So now the hydrophilic side can bind to whatever it needs to bind to absorb it or break it down and whatnot. Okay, cool. Protein digestion, yeah. So proteins are digested by enzymes called peptidases, yeah. And there's three types of peptidases. You need to know about these, yeah. So endopeptidases, they hydrolyze peptide bonds between specific amino acids in the middle of a peptide, the polypeptide, yeah. So they move in between the polypeptide and they hydrolyze peptide bonds in between it, yeah, not on the outside. On the outside, you got exo, yeah. Exopeptidases, they hydrolyze bonds on the ends of the polypeptide. So they come to the end of the polypeptide and they start hydrolyzing the bonds over there. Okay, cool. Dipeptidases, they break dipeptides, so two monopeptides that are put together, you make a dipeptide. They're gonna break that down into the individual amino acids that are made from them, yeah. Alright, cool. Lipid digestion and absorption details, yeah. So I'm gonna go through that. So this is the more detailed version of lipids, yeah. I'm gonna cover everything here, yeah, because I want you man to get obviously the maximum amount of marks, yeah. So you can use this information. I'll tell you which information is required and whatnot, yeah. Okay, cool. So before absorption can occur, yeah, fats must be broken down into smaller droplets to increase their surface area. We talked about that, yeah. So this process is facilitated by bile salts or bile acids, yeah, which are produced by the liver and stored in the gallbladder. Okay, cool. Bile acids help emulsify fats, turning them into tiny droplets called micelles, yeah. Nice. So you need to know that word micelle, yeah. Now we're gonna go into it, yeah. Once emulsified, lipids are acted upon by enzymes called lipases, yeah. Lipases break down triglycerides into their component fatty acids and glycerol molecules, yeah. This breakdown occurs at the surface of the micelles. So it's gonna all that's happening here is you got the micelle. It needed to become a micelle to increase the surface area. So now the enzymes, the lipases, yeah, they can act on that fast, yeah, and break it down obviously into triglycerides and fatty acids, yeah. Now, these triglycerides and fatty acids need to be reformed so they can be absorbed and therefore they can be used, yeah? So the micelles containing the breakdown products of lipids, yeah, move towards the surface area of the intestinal lining where they meet specialized cells called enterocytes, yeah? You don't need to use the word enterocyte, that's a bit extra. You can just say epithelial cell, yeah? That's fine. but you need to know the process here, yeah? So the micelles, they move towards the epithelial cell, and yeah, once they reach it, the fatty acids and monoglycerides that obviously got broken down from the triglyceride, they diffuse into the epithelial cell where they reassemble into triglycerides. So it's going to move into here, and using the Golgi apparatus and the smooth endoplasmic reticulum here, it's going to get made back into triglycerides. The difference here is it needed to be broken down so it can move across. So it was triglyceride first, it got broken down by lipase here, then it's going to move into the epithelial cell, and it's going to be reformed into a triglyceride here. Now, inside the enterocytes, newly formed triglycerides are combined with cholesterol and proteins and other lipids as well to form larger particles called chylomicrons. Yeah, you need to know that name. You need to know that word chylomicron. Yeah, it's required. Yeah, and these chylomicrons, yeah, they're then released into the lymphatic system through lacteals. Yeah, you need to know that as well. They're moved into the lymphatic system through lacteals, which are just vessels. They're just like channels that it can move through. So, little recap here. So the micelles come through. They have the fatty acids and monoglycerides. They combine with cholesterol and proteins and shit with the endoplasmic reticulum, yeah? Therefore, the micelles then move across to the lacteal where they get released, the chylomicrons, yeah? Not the micelles, the chylomicrons, yeah? The chylomicrons move through, and then the chylomicrons obviously go into the lacteal. And now the lacteal, they travel through the lymphatic system until they reach larger lymphatic vesicles called lymphatic ducts, yeah? You need to know that as well. Lymphatic ducts, yeah? These are just large lymphatic vessels, yeah? Which eventually empty out into the bloodstream. So then the chylomicrons, once they move through these lymphatic ducts here, they eventually reach the bloodstream here, which is near the heart. And once in the bloodstream, chylomicrons deliver lipids to various tissues throughout the body where they can be used for energy or stored for later use here. Okay, cool. So that's lipid absorption, yeah. So just make sure you know all of that, and you'll be sturdy with it. Okay, cool. Hemoglobin, yeah. Hemoglobin is a water-soluble globular protein, yeah, which consists of two beta polypeptide chains and two alpha helicases, yeah. So has two alpha helix, two beta chains, yeah, four chains in total, and it's also got a prosthetic group here, the heme group over there, which contains iron. So it carries oxygen in blood as oxygen combined to the heme group, the Fe2 plus group. Each molecule can carry four oxygen molecules, obviously, because you've got four heme groups, yeah. Okay, cool. Affinity, yeah. So affinity is like how much the hemoglobin like wants to bind to the oxygen, yeah. So if something's got high affinity, it's binding to the oxygen a lot. If it's got low affinity, it's not going to bind to the oxygen, yeah. So the affinity of oxygen for hemoglobin is influenced by the partial pressure of oxygen, yeah, which means oxygen concentration. So partial pressure, yeah, don't get confused by that. If it's got high partial pressure, it means there's better oxygen. That's it. That's the only thing it means, yeah. So when the partial pressure of oxygen is high, such as in the lungs, hemoglobin binds oxygen tightly, a process known as loading, yeah. So in the lungs here, there's better oxygen here, which means that it can prioritize on that and it can maximize how much oxygen it binds to. So obviously it's going to have a high affinity, so it can bind to oxygen tightly, yeah. This is called loading, yeah. Okay, cool. As cells use up oxygen, yeah, during respiration, the partial pressure decreases. Obviously, there's less oxygen, yeah, so therefore there's less oxygen concentration, which reduces hemoglobin's affinity. There's no need for oxygen to for hemoglobin to be binding to oxygen that much because oxygen is getting used up now, so it can unload it, it can leave it, yeah. So consequently, oxygen is released in tissues where it's needed, yeah. So it's unloaded. After unloading, yeah, hemoglobin returns to the lungs to bind with oxygen once more, which completes that whole cycle of loading and unloading, yeah. So as you see, O2 is loaded in the lungs here, and then it's released, and then unloaded in tissues, yeah, over there. You see, so these are the heme groups. They attach the oxygen, they have it now. Then they come back and they unload it into the tissues, yeah. Okay, cool. So you get these dissociation curves, yeah. And dissociation curves illustrate the change in hemoglobin saturation as partial pressure changes. So they show you like how saturated hemoglobin is compared to how if depending on how much oxygen there is in the situation. So obviously, if you got more oxygen, you're gonna have more saturation. It's gonna bind to it more. So these curves always increase like that. They always increase, yeah. But it just depends how they increase. How they increase shows like information about the organism, yeah. So the saturation of hemoglobin is affected by its affinity for oxygen, yeah. Therefore, in the case where partial pressure is high, hemoglobin it has high affinity for oxygen, of course, and is therefore highly saturated, and also vice versa, yeah. So let's talk about the inductive effect here. So after hemoglobin binds to the first oxygen, yeah, its affinity for oxygen increases, yeah, due to a change in shape. So when oxygen comes here, it binds to hemoglobin, it changes the shape slightly, which causes the affinity to increase. It's a shape that's more favorable, yeah, to attach to oxygen. So this change in shape makes it easier for subsequent oxygen molecules to bind. So because of the first one binding, it makes it easier to bind to the other oxygens, yeah? So this is known as cooperative binding, yeah? Dissociation curve explained. So initially the curve is always shallow, yeah, because it's hard for the first oxygen molecule to bind to the hemoglobin. It's not that easy, yeah. But once the first one binds, because of this positive cooperativity, yeah, it's therefore going to change the shape, making it easier for oxygen molecules to bind, yeah. Hence, a steep increase is shown in the curve, yeah. There's always a steep increase in the curve, yeah. This is called positive cooperativity, yeah. And finally, the gradient begins to flatten out again because the likelihood of the fourth oxygen binding is quite low because it's not going to find the binding site since already three of them are used up. It's got to find that final heme group here, which is obviously difficult to find. Okay, cool. Let me talk about the Bohr effect here. So the Bohr effect. So okay, cool. During cellular respiration, tissues produce carbon dioxide, obviously as a waste product. Yeah. So after respiration, it takes in oxygen, it leaves carbon dioxide. Yeah. So carbon dioxide diffuses into the bloodstream, yeah, and reacts with water to form carbonic acid, yeah. Carbonic acid, obviously weak acid, gonna dissociate straight away into hydrogen ions and bicarbonate ions, yeah. Okay, cool. So it dissociates into that. Obviously, we've got an increase in H plus concentration. H plus concentration increase makes something more acidic, obviously, naturally, yeah. And so hence the production of hydrogen ions from the dissociation of carbonic acid, yeah, leads to a decrease in blood pH. If something decreases pH, it makes it more acidic, like pH 2, 3, or 4. You get me? Cool. So it makes the blood more acidic now. Do you see that? So because it's more acidic, the decrease in pH caused by increased concentration of H plus here results in a conformational change in hemoglobin. When you got like increased hydrogen ions here, it normally changes proteins, like changes the way proteins are shaped here. So because of that, it's gonna create a change in the hemoglobin molecule, yeah. That shape change is obviously gonna cause a lot of situations. It's going to reduce the affinity of hemoglobin for oxygen, yeah? So this change in shape means that oxygen, hemoglobin, doesn't want to bind with oxygen as much anymore, you see, making it easier for oxygen to be released from hemoglobin into surrounding tissues. Because obviously, if you're making bare carbon dioxide, yeah, it means that respiration was already done. So we need to replenish the cells with more oxygen, yeah? So therefore, what happens is because the CO2 got released, it dissolved in the water, made carbonic acid, yeah? That dissociated into H+, increased the acidity of the blood, yeah? The increased acidity of the blood changed the hemoglobin shape, which therefore made it unload more oxygen to replenish the cells. You get it? Okay, cool. As a result of the Bohr effect, hemoglobin releases more oxygen to the tissues that are actively respiring and producing carbon dioxide. Yeah, this allows oxygen to be more efficiently delivered to cells where it's needed for cellular respiration. So this is called the Bohr effect. Yeah. Okay, cool. Now you need to know about hemoglobin in different situations. So for fetal hemoglobin, yeah, so that's obviously like a baby and whatever, fetus, yeah. So fetal hemoglobin has a higher affinity for oxygen, yeah, compared to adult, yeah. Obviously there's little oxygen inside inside the womb and that. So therefore, it's gonna need a higher affinity because it needs to be able to, clutch onto that oxygen more readily, yeah. This is crucial for the transfer of oxygen from the mother's bloodstream into the fetus across the placenta, yeah. Okay, cool. The dissociation curve for fetal hemoglobin is shifted to the left compared to that of the adult hemoglobin, yeah, indicating that at a given partial pressure of oxygen, fetal hemoglobin binds oxygen more tightly, reaching higher levels of saturation. So what you need to know is that for fetal hemoglobin, yeah, the dissociation curve gets shifted to the left. If it's getting shifted to the left, that means, yeah, that a higher point on the curve is going to be reached quicker. So what it means is that it's basically looking like steeper pretty much, yeah. So for the same amount of oxygen, yeah, it's gonna be more saturated. It's gonna bind onto it better, yeah, because it's got a higher affinity in general. They spawn with that. They spawn with a higher affinity. You get it? Okay, cool. Llama hemoglobin as well, yeah. So llamas also spawn with a higher affinity. Obviously, because they're in high altitudes, yeah, there's not much oxygen up there. So therefore, they got adapted hemoglobin with a higher affinity for oxygen to facilitate oxygen uptake in low oxygen environments, yeah. Calm. So therefore, the dissociation curve is the same as the fetal one. It's moved to the left, yeah, compared to humans, indicating a higher affinity for oxygen at a given partial pressure, yeah. Calm. Earthworm, yeah, obviously underground, there's not much oxygen, yeah. And also earthworms, they don't have like strong cooperative bonding as well, like humans do, yeah. Humans got stronger ones, these man don't, yeah. So again, lungworm or earthworm, lugworm, whatever that says, yeah, is gonna move to the left here compared to that. You get it? Okay, cool. Dove hemoglobin, yeah. So doves obviously they have high metabolic demands, yeah, because they gotta start flying and shit. They need to obviously, produce a lot of shit. So therefore they got a lot of reactions going on in their body, yeah. So therefore they need a lot of oxygen. Therefore, they have a higher affinity as well, since they need oxygen a lot more than humans do, yeah. and so therefore their blood also has a higher affinity for oxygen, yeah, allowing efficient oxygen delivery during flight. Hence, the curve is also to the left of a human's curve, yeah. Okay, cool. Circularity system, yeah. So large organisms require a circularity system because diffusion alone cannot efficiently supply substances like oxygen and glucose to the cells, yeah. So there's four things that you gotta have in a circularity system, yeah. Number one is obviously a suitable medium. So obviously blood serves as that medium since it's water based. It means that shit can dissolve into it easily, yeah. Number two is a means of moving the medium. So obviously animals have a heart, yeah, and the heart obviously pumps the blood around the system so that it can move and maintain the pressure differences, yeah. Number three is a mechanism to control flow. So we've got valves and veins, which obviously protect the backflow of blood, yeah. And lastly, yeah, you got to have a closed system of vessels. So the circularity system is typically closed and branched to distribute substances to all body parts, yeah. Alright, cool. So let me talk about a couple things here. First of all, in mammals, yeah, the circularity system is needed to be double, yeah. The reason why it's double is because one pump alone, yeah, is not gonna effectively push it around the body. So it's not gonna go from the like the lungs all the way down. It's gonna need to go through one pump here and then one pump backwards. You get me? Alright, cool. The heart acts as a central pump here with two chambers. One pumps the blood to the lungs for oxygenation, and obviously one pumps it to the body, yeah. The other one is a lot larger and it's a lot stronger, yeah, because obviously to move, blood from the heart to the body, it's gonna need quite a big pump, you get me? So there you go. Alright, calm. So effectively this is how it works, yeah. So obviously blood comes through, yeah. We've already established this here in part one, yeah, about the lungs and that. The lungs obviously oxygenate the blood, yeah, it turns red, yeah. Then it moves through the pulmonary vein, yeah, into the heart like that. Then the heart pumps it all the way down to the rest of the body, yeah. It gets used up in the organs, yeah, in the cells. Now it's not oxygenated anymore, so it's gonna come back through to the heart again. The heart's gonna pump that back to the lungs and it's gonna get oxygenated, yeah. So you see how it works. That's why it's a double system, yeah. Alright, cool. So to understand how the heart works, yeah, you need to understand the structure. You need to understand the picture that's painted in your head. What's going on? What's the situation? How can we break down what's in front of you? That's the real question, yeah, that you got to ask yourself. And the answer to that question, yeah, is that the heart is made up of two pumps here, each with two chambers, yeah. So what do I mean by these two chambers? I mean atrium, yeah. So you got right atrium there, left atrium there, yeah. Standard, yeah. Alright, cool. These are thin-walled and elastic, yeah. The atrium can stretch when filled with blood. Alright, cool. And you also got ventricles, yeah. That's the other chamber. So you got right ventricle there, left ventricle there. Simple, bro. Nice. Thick muscular wall to pump blood all around the body or to the lung, yeah. Because that's what's gonna pump it bang through the vein. You get me? Or through the artery, yeah. You get it? So that's what's gonna pump it like that. So it's gotta be very thick, very, you know what I'm saying, yeah. Say nothing, bro. Alright, cool. Between the atrium and the ventricles, there are a set of valves, yeah. So these valves are called the bicupsid and the tricupsid, yeah. Together, collectively, yeah, they're called atrioventricular valve, yeah, or AV valves is another name that you can call that. So the tricupsid is between my man over here, yeah, safe, and the bicupsid is between my man here, yeah, you get it. Okay, cool. Alright, nice. There are four main vessels that you need to know about, yeah. The first one is the aorta, yeah. So the aorta is right there. It's in that situation there, and it's connected to the left ventricle and carries oxygenated bloods to all parts of the body except the lungs. So what I'm saying here is it goes from left ventricle, the blood here gets pumped through the aorta, yeah, to the rest of the blood in it, to the rest of the body, fam. What am I talking about? Alright, cool. Pulmonary artery, yeah. Pulmonary artery, yeah, is connected to the right ventricle, so it's connected to the right one here, and that's gonna move it to the lungs, yeah. Obviously, the deoxygenated blood has come back from the body cells. It's lacking oxygen, bro. So it needs to get sent back to the lungs, so the lungs can refill that, replenish that. You get me? So it's gonna come back through like that, and then you get me. Yeah, it's gonna get sent up. So that's the pulmonary artery right there. Yeah, this blue thing here. Alright, safe. Pulmonary vein, yeah. So there's two of them. There's one there, one there. These are the pulmonary veins, yeah. This obviously is connected to the left atrium, and it brings oxygenated blood back from the lungs, yeah. So pulmonary means like you get me, it means lungs, innit? So it's gonna come back from the lung, bring my man into there for it to get pumped through the aorta and that. Alright, calm. Last one is vena cava, yeah. Vena cava is connected to the right atrium and brings the oxygenated blood back from the tissue, yeah. So back from the body when it gets used up, it's gonna come back through the vena cava. You get me? Back into the right atrium, yeah. Alright, say no more, bro. Let's talk about the blood flow, yeah? So you need to know where the blood is moving, yeah? If you want to become a specialist in the game, do you know what I'm saying? So first of all, yeah, it helps to understand where my man's moving from. So first of all, yeah, deoxygenated blood, yeah, comes into the right atrium through the vena cava, yeah? So deoxygenated blood has been used up inside the cells, yeah? It's going to come all the way back from the body, you know what I'm saying? From your leg, from your whatever. It's going to come all the way back through, yeah? And then bang into the vena cava. You get me? That's where it says number one. It's gonna come through there. Then it's gonna move, yeah, through the tricuspid, yeah. The tricuspid is that thing where it says four, yeah. It's gonna move through the tricuspid, yeah, from the right atrium to the right ventricle, yeah. Then it's gonna come through the pulmonary artery, yeah, and get sent all the way back to the lung, yeah, to get refilled. That's the one route, yeah. Second route, yeah, is oxygenated blood comes from the lungs, yeah. So once it's been oxygenated, it's gonna come back through eight. That's eight. That's the pulmonary vein, yeah, and it's gonna come into the left atrium, yeah, and then it's gonna move from left atrium where it says 9 through 10, yeah. That's the bicupsid, yeah, to 11, which is the left ventricle, yeah. And then it's gonna move. The left ventricle is gonna pump that. It should say left ventricle there, yeah, not left atrium. It's gonna pump that back through 12, yeah, which I'll talk about what 12 and 6 are in a minute, yeah. It's gonna move through 12 all the way back like that through the aorta to the rest of the body. You see that? You get the situation. So make note of that. You can also memorize it as it should switch in it. So it should go A V A V A V, whatever. You get me? What I mean by that is atrium, vena cava, then ventricle, but don't worry about that. And then pulmonary artery, yeah, and then pulmonary vein, and then atrium, and then you get me aorta, yeah. Alright, calm. Alright, safe. So another thing you need to know about, yeah, is I said that these are called AV valves, yeah, bicuspid and tricuspid, yeah. But these ones here that are inside the aorta and that, yeah, and pulmonary, yeah, is called semilunar valves, yeah. So this is SL ones, yeah. So these are AV, tricuspid and bicuspid are AV valves, yeah, and these two are SL valves, yeah, semilunar valves. Alright, calm. Now let's talk about how the heart actually pumps shit, yeah. So diastole, yeah, diastole, yeah, is to diastole means like to relax in it. So diastole means like relax, you get me? Systole means to contract in it, it means to start moving, start doing what it needs to do. So atria and ventricles, they relax, yeah, they start chilling out, they start baking off, you know what I'm saying? Then you get elastic recoil of heart, lowers the pressure inside the heart chambers, and blood enters to the heart from the vena cava and the pulmonary vein to fill the atria. So basically, what that means here is the atria and the ventricles, they start relaxing, they start chilling out. And what happens is blood comes through my man up here and fills up the atrium, and it comes through my man over here, the pulmonary veins here, and fills up the atrium. So oxygenated blood, deoxygenated blood, yeah, fills up atrium from the body, fills up atrium from the lungs. You get me? On both sides. Pressure increases in the atria, obviously, because the atrias are now getting more full and full, yeah, it's going to start filling up with pressure in it. So it's going to have a lot of pressure. So that pressure is going to open the valves in it. So it's going to open my man, yeah. And that's going to open the AV valves, yeah. Obviously, that's tricuspid, bicuspid, yeah. But together we can just call it AV, yeah. Atrioventricular, yeah. Alright, cool. The relaxed atria and ventricles means that the semilunar valves are closed, yeah. Because these are relaxed, they're not pumped, yeah. Think about it. If I pump these, yeah, it's gonna open my man. It's gonna open the SL where my mouse is, yeah. But because they're relaxed, yeah, they're not gonna, you know what I'm saying? Pressure's still kind of chilled out. It's not gonna do nothing, bro. Alright, calm. Atrial systole, yeah. So now what happens? I told you systole means to contract here. So now the atria then contracts. So the atria now filled up with bare blood here. It's got a lot of blood in there. And now what's gonna happen is it's gonna contract here. And when it contracts, it's gonna push all the blood through. And when it pushes all the blood through here, it's gonna move into the ventricle. So now all the blood is going to the ventricle, all the blood's going to the ventricle from there. Obviously that creates a little increase in pressure, yeah. So it's gonna open the SL valve slightly. Not fully, but slightly. They're beginning to open. You get that? Now we've got ventricular systole, which was when the ventricles pump here. That's when you hear that other pump here. That's going to be contraction of the ventricles, which causes the the AV valves to close here and the semilunar valves to open. That's allowing blood to leave the left ventricle through the aorta and the right ventricle through the pulmonary artery here. So now the ventricles contract here, which pushes blood from the ventricle through there and pushes blood through the ventricle through there, through the aorta, yeah, to the rest of the body. And this goes back to the lungs, yeah. You see the situation, so that's how it works, yeah. That's the heart cardiac cycle, yeah. So that's what happens, bro. And the reason you hear like two pumps when somebody breathes or whatever is because that's the valves closing in and opening. So that's a little summary there. So you got dystole, yeah. You got atrial systole, yeah, and then you got atrial, and then you got ventricular systole, yeah. So that's the stages there, basically. You just need to memorize that. Simple, bro. Arteries, yeah. So arteries, these are adapted to carrying blood away from the heart to the rest of the body, yeah. They are thick-walled to withstand high blood pressure, contain elastic tissue which allows them to stretch and recoil, thus smoothing the blood flow, yeah. You need to talk about that. You need to talk about how the elastic tissue smoothens blood flow, yeah. They like it when you use them kind of words in it. They also contain smooth muscle, which enables them to vary blood flow, lined with smooth endothelium, yeah, to reduce friction and ease the flow of blood. Cool. Say nothing, bro. Obviously, they got a pretty big lumen here, yeah, but not that big. You also got like a lot of muscular wall and that kind of thing, yeah. Cool. Obviously, to withstand the high pressure in there. Yeah? Arterioles, yeah, are just smaller versions of arteries, yeah, that lead off to the capillaries. You get it? So they branch off arteries, yeah. They have thinner and less muscular walls, yeah, and their role is to feed blood into the capillaries, bro. Alright, cool. Capillaries, we already like spoke enough about them, yeah. They're just like tiny vessels that obviously take, they're one cell thick, they obviously take, their site of metabolic exchange, which just means like, like oxygen intake and that kind of thing, yeah. Alright, cool. Veins, yeah. So veins have a much larger lumen, yeah, to because obviously they got a, you know what I'm saying? Yeah. That means the pressure is going to be low, yeah? So they carry blood from the body to the heart, yeah? And contain a wide lumen to maximize the volume of blood carried to the heart, yeah? They are thin-walled and blood is under low pressure and contain valves to prevent the backflow of blood, yeah? So obviously because they're like, they got low pressure, that means that the blood's not going to be moving in one direction. It's going to want to go back since it's moving so slowly, yeah? So therefore you got to have these valves in it to stop the backflow, yeah? Alright, cool. So there's little elastic tissue or smooth muscle as there's no need for stretching and recoiling, yeah. Since there's such like low pressure, yeah, there's no need for anything to withstand that pressure. That means that you can maximize the amount of blood that you're moving. You don't have to worry about like the smooth muscle or elastic tissue and all that, yeah. Alright, cool. Tissue fluid, yeah, tissue fluid, yeah. Alright, cool. So tissue fluid is a liquid. Do you see the little effects there? Yeah? Saying that. Alright, cool. Tissue fluid is a liquid containing dissolved oxygen, nutrients, and waste products, yeah. Um, is that mad? Say nothing, bro. Tissue fluid formation, yeah, so you need to know about that. So hydrostatic pressure, yeah, exceeds osmotic pressure, pushing the fluid out of the capillaries. So basically, what's going on here? What I mean by that is hydrostatic pressure is the pressure created from the heart pumping, yeah. So the heart's gonna be pumping, yeah, and osmotic pressure is the pressure that is yo, my voice just went mad, you know. Cool. The osmotic pressure, yeah, is the pressure that moves is what's like the pressure that's causing the water to like stay inside the capillary and not be moved out, yeah. So what's happening here, yeah, is the hydrostatic pressure, the pressure of the like the heart pumping, yeah, exceeds, yeah, the force that's trying to keep the tissue fluid back inside the capillary. So since the pressure of it to leave the capillary is greater than the one to keep it in the capillary, it moves out, yeah. So pressure forces tissue fluid out, and fluid contains dissolved oxygen, nutrients, and waste products, forming tissue fluid in interstitial spaces, yeah. Safe. So why do you need this? Why is it a good thing? Yeah, well, obviously it contains dissolved oxygen, yeah, which is essential for cellular respiration and energy production. Yeah, it also contains nutrients, yeah, such as amino acids, fatty acid, glucose, bro, which provides building blocks for cellular growth, repair, and metabolism. Yeah, alright, cool. Ions, yeah, regulate cellular processes such as nerve transmission and muscle contraction. Yeah, obviously. And tissue fluid facilitates the removal of waste products, including carbon dioxide and metabolic byproducts. Yeah, alright, cool. Reabsorption, yeah. So the larger molecules, they're still inside the capillaries, yeah. So all the little shit got forced out, yeah. But since the larger molecules are still in the capillaries, yeah, it wasn't enough pressure. They can't just move across the capillary like that. They're too big, yeah. It's gonna lower the water potential because deep it bare water left, yeah. And all the like the heavy shit is still inside, so it's still quite concentrated in there, yeah. Which means that the water potential is gonna get lowered, yeah. Therefore, water is gonna re-enter the capillaries by osmosis, obviously because it's got more water outside than inside. So it's gonna move from higher water potential to lower water potential, yeah. Water reabsorption helps maintain blood volume and prevents excessive fluid loss from the tissues because obviously if your fluid is drying out, your blood is going to be dead. You get me? Alright, cool. Lymphatic system, yeah. So eventually, yeah, equilibrium is going to be reached and some liquid will not be reabsorbed, yeah. To reabsorb all that liquid, yeah, or most of it, yeah, you can't do that because osmotic pressure is going to balance in it. So it's going to have the same water potential inside as outside, yeah. So that's why you got to do a bit of a situation here now. The lymphatic system is responsible for returning excess fluid back to the bloodstream, yeah? So the way it does this is there's lymph nodes, which filter lymph fluid, yeah, and remove bacteria, viruses, and other foreign particles. Obviously, this fluid has come from the cells now, since it got forced out the capillary into the cells. So it would have had all this madness around it, so it's got to come through the lymph node to filter all that out, yeah? Calm. Lymphocytes in the lymph node also play a crucial role in the immune defense, yeah? Safe. Vascular bundle, yeah? So vascular bundle in the roots here. So xylem and phloem are components of vascular bundle which enable the transport of substances as well as provide structural support here. So the xylem vessels are arranged in an X shape in the center of the vascular bundle. So only in the roots. And the roots is in like a little X shape here. And this enables the plant to withstand a lot of force in the roots because obviously it's going to be a lot of force from water and that kind of thing here. So it's going to have to like combat that. The X-shaped arrangement of xylem vessels is surrounded by the endermis here, which is an outer layer of cells which supplies xylem vessels with water. So that's where the water gets entered through is the endodermis here. We'll talk about the endodermis in a minute here. There's also an inner layer of meristem cells. Obviously, these meristem, they get moved into whatever cells you want them to be, yeah? Alright, cool. Vascular bundle in the stem, yeah? Bundle, yeah. Alright, calm. The xylem is located on the to provide support and flexibility to the stem. Phloem is found on the outside of the vascular bundle, yeah. So in the stem, yeah, the phloem is on the outside of the vascular bundle like that. They're inside. They're like inside. If you see that, they're like layered inside the thing, yeah. And so xylem is there, and the phloem is there, and there is a layer of cambium, yeah, in between the xylem and phloem, which are meristem cells involved in the production of new xylem and phloem, yeah. So that's the situation there. You got these things around it, which is gonna help, make new xylem, new phloem cells, yeah, because they're meristem in it. Alright, safe. Vascular bundle in the leaf here. So the vascular bundle forms the midrib and veins of a leaf here. So this thing here is the midrib that comes out like that, and these are the veins that are coming out here. And leaves have a network of veins starting at the midrib and spreading outwards here, which are involved in transport and support here. Safe. Transpiration. So this is the process where plants absorb water through the roots, which then moves up through the plant and then it's released to the atmosphere as water vapor through pores in the leaf here. So we need to talk about how that's done. First of all, yeah, the xylem, yeah, you need to understand what is the xylem. They say up the xylem, down the phloem, yeah, but what does that actually mean? So the xylem, they transport water and minerals and provide structural support, yeah. They are long cylinders made of dead tissue with open ends, therefore they can form a continuous column, yeah. So you need to know about that. They're made of dead tissue, yeah, so that maximizes the efficiency in between and stuff like that. Maximizes the space as well, yeah. Xylem vessels also contain pits which are enable water to move sideways between the vessels, yeah. So they have these kind of pits, which means that it can move sideways between the vessels and inside and whatever it needs to do, yeah. They are thickened with a tough substance called lignin. You need to know about that, which is deposited in spiral patterns, yeah, to enable the plant to remain flexible. So this spiral shit is the lignin, yeah, and that's what, what I'm saying, thickens it up here. You need to know about that. You need to know that it's a spiral as well. Transpiration stream. So the transpiration stream, yeah, is the movement of water up the stem. It enables processes such as photosynthesis, growth, and elongation as it supplies the plant with water, which is necessary for all those processes, yeah. The transpiration stream also supplies the plant with the required minerals whilst enabling it to control its temperature via evaporation of water. Since, at the end of the transpiration stream, it's going to leave through the leaves, it's going to get evaporated, which means that it's going to, cool the plant down, obviously, yeah. Calm. Calm, bro. Overview. So, transpiration involves osmosis, obviously, yeah, where water moves from the xylem to the mesophyll cells, yeah. Transpiration also involves evaporation from the surface of the mesophyll cells into intercellular spaces here, and the diffusion of water vapor down and water vapor potential gradient out of the stomata. So there's two ways that water is evaporated, yeah. First of all, yeah, is evaporation of water from the surface of the mesophyll cells to the airspace, yeah. So that's number one here. So they go from the mesophyll cells, yeah, and they evaporate through the air spaces and eventually leave. You get it? That's what I was talking about here, yeah. Number two, yeah, is diffusion of water vapor, yeah. So since there's a lower concentration of water vapor outside than inside, it's just gonna move through the stoma via, what I'm saying, osmosis effectively or diffusion. You can argue, yeah. Alright, calm. Portometer, yeah. Portometer is investigates the rate of transpiration and can be investigated with the help of a portometer, yeah, where water lost by the leaf is replaced by water in a capillary tube. So obviously you got some water in a capillary tube like that. Water's gonna get lost by my man, and this air bubble is gonna move, and so you can measure it on the ruler in it. So the movement of the meniscus or bubble can be used to determine the rate of transpiration. Yeah. Safe. Factors that affect transpiration rates. So obviously the number of leaves, the number, size, or position of stomata, the presence of a waxy cuticle, the amount of light present, the temperature, and the humidity of the air, and also the water availability. Yeah. Cool. Xerophytes here. So they have smaller leaves here. That's an adaptation of them. You need to know about the adaptations of xerophytes here. So they got smaller leaves to reduce surface area for water loss. They're also densely packed mesophyll, yeah, and thick waxy cuticle to prevent evaporation. Densely packed means there's less air spaces, which means less evaporation you get. And also the cuticle is going to stop evaporation in it. Closure of stomata in response to low water availability. They got presence of hairs and pits to trap moist air, reducing water vapor potential gradient. And leaf rolling to minimize exposure of lower endodermis to atmosphere, trapping moist air, yeah? Alright, cool. Okay, so let's talk about the path of water through the root, yeah? So, water enters the root hair cells and moves into the xylem tissue. Calm. So it enters through the root, yeah, and it moves into the xylem, yeah? So it's going to enter the root here and to the xylem, but it's going to need to cut through all of these first, yeah? So this movement occurs because of the water potential gradient, as the water potential is higher in the soil than in the root hair cells, yeah? So obviously there's more water in the soil compared to the root hair, so it's going to move in through osmosis, yeah? Alright, calm. The root hair cells provide a large surface area for the movement of water to occur. Standard, bro. Minerals are also absorbed through the root hair cells by active transport, yeah? Calm. So simplest pathway, yeah? So you need to know about simplest pathway, yeah? So water enters the cytoplasm, yeah? Through the plasma cell membrane and passes from one cell to the next through the plasmodesmata, yeah? The channels which connect the cytoplasm of one cell to the next. So what this means here is simplest pathway is basically just water moving through the cytoplasm, yeah? So the water needs to move through the cytoplasm like that, and it does it through these little things here called the plasmodesma, yeah? So that thing there, that's hiding it, you get me? So that's what it means. So simplest, yeah, is just going straight through, so just through the cytoplasm, you get it? Apoplast, yeah, is when it goes through the cell wall. So water moves through the water-filled spaces between cellulose molecules and the cell wall. So obviously cell wall is made out of cellulose, yeah, which obviously has gaps here, so that water like comes through the cellulose here, moves into those gaps inside the cellulose, into like the cracks in the wall effectively, yeah, and it just cuts through the cellulose direct. In this pathway, water doesn't pass through any plasma membranes, therefore it can carry dissolved minerals and ions and salts here. So for the other one, it's got to move through plasma membranes, it's moving through cytoplasm here. Apoplast here is just moving through the cell walls, so it means it can move a lot more shit with it. You get it? And then here you got the Casparian strip here in the endomus here. So I'm gonna talk about that. So Casparian strip here. So when the water reaches a part of the root called the endomus here, it encounters a layer of suberin here, which is known as the Casparian strip here. Suberin is just a type of, you know what I'm saying, polymer that's present inside of there, yeah, which cannot be penetrated by water, yeah. Whereas cellulose can be penetrated, but that shit can't be, yeah. Hence, the water that has been moving through the cell must now enter the simplest pathway, yeah, to cross the endomus, yeah. So obviously, it was moving through apoplast first, yeah. Apoplast means moving straight through the cell walls, yeah. But now it can't do that since the shit's impenetrable, yeah. So it means it's got to start moving through the cytoplasm now, yeah. So simplest pathway, yeah. So once it has moved across the endomus, the water reaches a pit in the xylem vessel, which is known as the entry point of water. Alright, calm. Now, water movement up the xylem, yeah. So the push of water upwards is aided by the root pressure, yeah, that you need to know about that. There's root pressure, which means that it's gonna, push the water up the xylem, yeah, which is where the action of the endomus moving minerals into the xylem drives water up the xylem by osmosis, yeah. So that's what root pressure is, yeah. It's the it's the way that minerals move into the xylem from the endomus, yeah, which obviously creates pressure, osmotic pressure, yeah, or you can call it root pressure, which is gonna drive water by osmosis into the xylem, yeah, which is gonna push it upwards, yeah. Alright, cool. The flow of water is also maintained with the help of surface tension and cohesion. Yeah, so obviously water particles they because of the hydrogen bonds here they start sticking together. That's called cohesion. Obviously, you know about that from topic one. So that's gonna start moving it as like a little chain up the xylem. Yeah. Alright, cool. The action of these two forces in combination is known as the cohesion tension theory, which is further helped adhesion to the walls of xylem. Yeah. It's further helped by adhesion, yeah. So adhesion is when water molecules don't stick together, they stick to surfaces, yeah. So they stick to the side of the xylem, yeah. So the water molecules are sticking together and they're sticking to the side of the xylem. So therefore it's gonna move up the xylem logically, yeah. You get it. Safe. Translocation. So translocation is a bit of a long topic, yeah. But yeah, I include everything in it because I want you to get max marks here. So obviously you need to know all this. So yeah, calm. Translocation, yeah. So translocation is the way that sucrose is gonna move from the source cell, yeah, which is the leaf cell, to the sink. The leaf here is where obviously sucrose is gonna be made, yeah, from photosynthesis, yeah, and it's gonna need to move straight down to the to where it needs to be used up. Obviously, sucrose is gonna be used up in different parts of the cell, yeah, like the root cells, the other kind of cells, any cells that are using it, yeah, and also water is also gonna be moved at the same time. So translocation, yeah. So sucrose enters a phloem in a process known as active loading, yeah, where companion cells use ATP to transport hydrogen ions into the surrounding tissue, yeah. So sucrose is gonna enter the phloem like that through active loading, yeah, which is where the companion cells, my man over here, yeah, is gonna use ATP to transport hydrogen ions into the surrounding tissues around it. Hence creating a diffusion gradient, which causes the H plus ions to diffuse back into the companion cells, yeah? So that maintains like a diffusion gradient. You might be asking, what was the point of it moving outside the companion cell if it's gonna move back in, yeah? You're looking at it the wrong way. It's meant to move the H plus outside, but the reason that it's moving back in is that it still creates like a chain, like a cycle. So now when it moves back in, it can move back out again, bro, by ATP. If it would just move back out, the cycle would end, you get me? So some need to diffuse back in, yeah? So it creates a diffusion gradient, yeah? So it moves the H plus outside, which means there's more H plus out than in. So it's going to move back in slightly, yeah? But obviously because active transport is still moved out, yeah? Then facilitated diffusion, yeah, involving the co-transport of proteins, yeah, allows a returning H plus ions to bring sucrose molecules back into the companion cells, yeah? So When it moves out, yeah, it's gonna move the sucrose back into the companion cells, yeah, because it's gonna go through the co-transporter protein, yeah, so like co-transport, and the returning H plus ions, they're gonna bring the sucrose back into the companion cell, yeah. Alright, cool. So you can imagine that. So that's why sucrose is moving that way there into the companion cell, because obviously my man got forced out. The H plus is gonna return back into there using the sucrose, and it's gonna bring the sucrose with it, yeah. So now the sucrose is inside the companion cell, yeah. Hence the concentration of sucrose in the companion cell increases, yeah. As a result, the sucrose As a result, yeah, the sucrose diffuses out of the companion cell down the concentration gradient into the sieve tube elements, yeah, through links known as plasmodesmata, yeah, the thing from earlier, yeah. So what's happening now is because the sucrose got moved from the leaf cell into my man, yeah, since there's a lot of sucrose here, it's gonna move down the diffusion gradient, yeah, into the phloem, yeah, but there's little parts inside the phloem here called the sieve tube elements, yeah, that little white part there, you see it before you get to the phloem, yeah. So once it goes into there, it's gonna build up inside of there. You get me? As sucrose enters the sieve tube elements here, the water potential inside the tube decreases. Obviously, because you've got more like concentrated shit, so the water is gonna decrease and the water potential is gonna decrease here. Therefore, causing water to enter via osmosis from the xylem. So because the water potential is decreasing here, yeah, water is gonna enter through from the xylem straight directly into the phloem like that. You see the situation because the water potential is decreased over here. Yeah? That increases the hydrostatic pressure of the CF tube element, yeah? As a result, water moves down the CF tube from an area of high hydrostatic pressure to area of low hydrostatic pressure, yeah? Eventually, sucrose is removed from the CF tube elements by diffusion, active transport into the surrounding cells, increasing the water potential in the CF tube, yeah? Hence, water leaves the CF tube by osmosis back into the xylem, and as a result, reduces the pressure in the phloem at the sink here. So that's what's going on here. It's going to move down, the water's going to move like that, and obviously it's going to move back into there. Obviously it's going to move it back into the xylem since the pressure is decreased now, and obviously the sucrose is going to be moved into the sink cells, yeah, where it needed to be moved. Alright, calm. Evidence here. So there's a pressure in the CF tube elements here, obviously because the hydrostatic pressure is going to increase in there and whatnot, yeah, as shown by sap being released when the stem of the plant is cut, yeah. So when you cut the plant, yeah, the sap is like being released, yeah, in a fast way. That means that there's clearly like a lot of pressure inside the CF tube, yeah. So that's evidence for mass transport, yeah. The concentration of sucrose is higher in the leaves, yeah, the source of the plants, than in the roots, yeah. That's obviously because it's going to move from the top to the bottom, yeah. So it's going to move from high hydrostatic pressure to low hydrostatic pressure, yeah. So it's natural that the concentration of sucrose is higher in the top, yeah. Increases in sucrose levels in leaves are followed by a similar increase in sucrose concentration in the phloem, yeah. So increase in sucrose level in the leaves are followed by a similar increase in sucrose concentration in the phloem. So what this means is that naturally, because that's the way the process goes, yeah, it goes from the source cell into the, you get me, into the phloem, yeah. That means that naturally one follows the other one. You see what I'm saying? Metabolic poisons inhibit translocation of sucrose in the phloem. So because of the way it works, yeah, if there wasn't these poisons, yeah, they wouldn't inhibit translocation if it didn't work like that in that kind of way. Because it works like that across the phloem, yeah. If you have something that poisons the phloem, it's not gonna move. Alright, calm. Criticism, yeah. So the function of the CF plates is unclear, as they would appear to slow mass flow. Obviously, what a certain man argues that there's no need for that CF plate because it can just go directly into the phloem. However, a certain man suggests that they have a structural function to help prevent bursting under pressure. Not all solutes move at the same speed as well, which is, if it was mass flow, if it was correct, this whole theory, yeah, then it should be moving, they should all be moving at the same speed, yeah. Sucrose is delivered at the same rate to all regions instead of going more quickly to the ones with the slowest sucrose concentration, yeah. So in real life, sucrose is obviously delivered at the same rate to all places, yeah. If it worked the way that this works, yeah, it should go to the places with the lowest pressure, yeah, the lowest sucrose concentration first, since it's gonna move from the highest to lowest faster, yeah. But that's not the case in real life, obviously, yeah. Cool. Now there's evidence for this here. There's evidence against the criticism here, which is ringing experiments here. Ringing experiments here, they investigate if the phloem is responsible for mass flow, yeah. So what you do is you remove the bark, yeah, and the phloem of the tree, which means that it's just the xylem. So, and you see what happens here. So over time, the tissues above the missing ring here swell with sucrose solution, and the tissue below dies. This therefore shows here that sucrose is transported in the phloem, yeah, because once you remove the phloem, it no longer got transported. You see that it starts staying inside the top and the top here, and it starts staying there. You get me? So you'll see it here. The bark and the phloem got removed, yeah. So now the sucrose just swells up inside there, and everything below that dies because it can't move across. So that proves that the phloem has something to do with it. You see what I'm saying? You also got tracer experiments here. Tracer, and everything's like called tracer, it means it's radioactive in it. So Tracer experiments can also be used to investigate the transport of sucrose in plants. Plants are grown in an environment that contains radioactivity, labeled carbon dioxide. Yeah, obviously CO2 is needed to make sugar. Yeah, you need to make sucrose. Yeah, so therefore in photosynthesis, if the CO2 is radioactive, that means that the sugar is going to be radioactive. Yeah, so this means it will be in the sugar produced in photosynthesis. Yeah, the movement of these sugars can now be traced through the plant using autoradiography. Yeah, so you use this autoradiography here to start looking at where that sugar is, yeah, and you can trace it where it is in the plant, yeah. And the areas that were hit by radiation from the sugars will be black. So the areas on this like radiograph here, they'll be black if that's where the radiation is. And those areas are obviously the phloem. So that proves that the sugar is inside the phloem, and that proves that the phloem transports sugar, yeah. Calm. And that's the end of the whole topic, yeah. So it's a bit of a long topic still, I can't lie. Yeah, it's dead one, but man, calm.